A very good morning to everyone. Welcome to Amity International Societies for Natural Products webinar series. And today we have a very eminent, world-renowned professor, Professor David Morales. And on behalf of our Honorable Founder President, Sir, Dr. Ashok K. Chauhan, our Chancellor, Sir, Dr. Atul Chauhan, Sir, Vice Chancellor, Ma'am, Dr. Balvinder Shukla, Ma'am, from President ASTIF, Dr. Selvamurthy sir, Assistant President ASTIF, Vice President ASTIF, Group Captain Shukla sir, from Amity Institute of International Alliances, DG and Honorable Scientific Advisor to Founder President Dr. Rajiv Sharma sir, from Dean Health and Allied Sciences, Dr. BC Das, from Dr. Ajit Nagpal sir, who is Vice President for RBEF, Ritanand Baldev Education Foundation, and he also looks after the Middle East operations of Amity University. From Additional Chancellor Sir, Dr. Asim Chauhan Sir, and whole scientific fraternity and all the scientists who have joined us from India and abroad, the students, we welcome you all to this webinar series. And before I request Professor Dr. David Morales to give his presentation, I will just read out his brief resume. It's like world connecting at one place. So Professor Morales carried out his BSc and MSc in chemistry at the School of Universidad Nacional Autonomía de Mexico, UNAM, and was awarded his PhD from Essex University, UK in 1998. He is beneficiary of the Cognac Reciprocated program after 3 years of postdoctoral fellow at the University of Hawaii at Manoa, USA. Professor Morales takes his current position at the Instituto de Química, UNAM, Mexico. So Professor Morales has graduated over 110 BSc, MSc and PhD students and published over 170 papers, including 15 reviews in top JCR journals of its specialty, as well as 11 book chapters. He has served as editor-in-chief of the books The Chemistry of the Pincer Compounds in 2007 and Pincer Compounds, Chemistry and Application in 2018, which was edited by Elsewhere in Netherlands. And he has also co-authored the textbook on Organometallica, edited by Chemistry School of UNAM. These publications have been cited over 4,800 times for an H index of 33. And he belongs to the Sistema National de Investigadores in the highest level, that is level 3, and to the Pride UNAM in the highest level of UNAM, that is level D. He is member of chemistry societies in Mexico, Spain and USA, and has been visiting professors at universities and research centers in Spain, Germany, Netherlands, and Sweden, as well as different campuses of the University of California, that is Riverside, San Diego, and Santa Barbara. He performs reviewing activities of manuscripts of more than 50 chemistry journals of the most influential editorial houses in the world, such as Elsevier, ACS, RSC, ARCAD, Bentham, Wiley, Taylor and Francis, Springer, Hindway, and has been reviewer for different international funding agencies such as for Technologica, Contact Mexico, Petroleum Research Fund USA, National Science Foundation USA. He has been into Fondo National Sintioia, Technologica, that is Fondicit Child, the Czech Science Foundation of Czech Republic, Israel Science Foundation of Israel, the ECOS Contact of France, Mexico, ECOS Fondiat France Child, Aegis National de la Richards, that is France, National Research Foundation South Africa and of Argentina, and the Executive Government Agency of National Science Center, Polonia. So I think you have covered the entire world in research funding. It is It has represented over 100 invited conferences in USA, Venezuela, Spain, Netherlands, Portugal, Germany, UK, Sweden, China, France, Russia, Poland, Canada, Japan, Paraguay, and Mexico. And he has presented over 500 papers in national and international conferences and symposia. By invitation, Professor Morales belonged to the editorial board and referees for the Journal of Ariquak, of the International Advisory Board of the Journal Chemistry, Catalytic Chemistry of Willy, the editorial board for the Journal of Inorganic Chemistry, and of EC Chemistry, the editorial board of the Journal of BAOG, of Current Journal of Applied Science and Technology, Advanced Research in Chemistry and Applied Science, SN Applied Science, and in African Journal, Organic and Medicinal Chemistry, Inorganic Chemistry, Current Organic Chemistry, and Clinical Drugs of Bentham. Then second editor for the Journal of Current Organic Synthesis by Bentham and of the International Advisory Board of the Homogeneous Catalysis Symposia. He has served as the guest editor for Inorganica Chimica Acta and of molecules from MDPI. He has also been recognized as one of the most cited authors in chemistry at the UNM Mexico consequently in the years 2012, 13, 14, and has been awarded by DGAPA, UNM, and also by Elsevier Scopus. He has served as chairman of the meeting Polymath, continuously in year 2015, 17, and 19, which was an international conference about polymers and advanced material. He has been organizing committee of the U Centro and Quimica Inorganica and N Centro di Quimica Inorganica at the International Conference of Coordination Chemistry, and he has been also the chairman at the first international conference on pincer chemistry held at Mexico. So with this introduction, and we are also joined by our eminent panelist, Dr. Nagpal sir, who looks after the complete Middle East operations of Amity University. I request you, Dr. Morales, to kindly, begin your webinar. A very warm welcome to you from the Amity fraternity, and we are eagerly looking forward to listen to you, and kindly share your screen and presentation. So welcome, Dr. Morales. Yeah, thank you very much. Yeah.
Thank you very much, Asha. Thank you very much for the kind invitation. Let me just share the screen for the presentation, and we will start with this. So, as I said, thank you very much for the kind invitation. I think it's a very nice way that we have actually learned that we can be connected, and of course, we avoid the hassle of the travel, but we also don't enjoy the traveling and the fact that we can actually meet together. But at least for the moment, we can actually do this thing in a virtual manner. So once again, thank you very much for the invitation. And today I'm going to be talking a little bit about a combination of organic ligands, and this is going to be also very interesting, and some of the research we're actually doing with natural products that we are trying to isolate these compounds and trying to put also metals. So trying to enhance in that way the overall the medical possibilities that we can actually have with those compounds. For most of the things that we do, generally these fall into two different areas. One is coordination chemistry, and the other one is organometallic chemistry. So generally, at least for us here in Mexico and generally around the world, this takes for a couple of semesters, perhaps. There are two different courses, one for coordination chemistry and another one for organometallic chemistry. And I'm going to try to explain both areas to you guys this time in a couple of slides. So just to give the main difference and just to show you why this is actually very interesting. And they have very nice applications in medicinal chemistry and several other applications in biomedical sciences and stuff. So, think of as we were actually talking with Professor Carwell, These are some of the contacts that we have. If you have any question after the talk, this is my email, my personal email, and this is the web page where you can actually see some of the other research that we actually do, not necessarily based on medicinal chemistry or biomedical applications, but also some of the other applications that you can actually have with these compounds in catalysis, for instance. So first of all, because sometimes I just run out of time, I just wanted to thank, this is most of the research group that we have in Mexico. This is, of course, this lady that is very happy because she's getting rid of us because she has already got her degree. But the other ones are actually very enthusiastic, a bunch of guys that they are doing all the work in the lab, and they share a lot of experiences, and the experience that these guys actually bring into the lab is very important for us. So, and this is actually the crest of our university. This is a grand saloon that where we actually carry the bypass or the defense of the thesis. So let me start with this guy. The name of this guy was, at the time, he was Dr. Alfred Werner. He was situated at Switzerland, and we are talking about, this is the beginning of the 20th century. Alfred Werner is known as the father of the coordination chemistry, particularly in Europe, because there is another guy who was very important in the Americas. But yeah, in general, I mean, this guy was the one who was starting with all the explanation regarding coordination chemistry. Coordination chemistry is something very simple to understand. It's very basically that we can actually have organic molecules. Generally, these organic molecules, they have a moiety, they have a skeleton that is composed by generally hydrocarbon backbones, and these hydrocarbons generally they have donor atoms. Generally, for instance, we prefer phosphorus, but it can be phosphorus, it can be nitrogen, it can be sulfur, it can be oxygen, very basically any other donor atom that it can actually provide with electrons, electron pairs to actually donate to a given metal. So the explanation that Alfred Werner gave to understand coordination chemistry, all the chemistry that he actually did, awarding him the Nobel Prize in 1913. So this is already more than 100 years. And this logo that we have over here, that you can actually see, it says 100 años in Spanish. It's 100 years. It was a celebration that we was a few years ago at the chemistry school here at the National University in Mexico. So this was actually to commemorate these achievements, because I mean, this guy not only actually gave an explanation to understand the way they interact, the metals, with some organic molecules, but also open a big window, a big saloon, I would actually say, to possibilities in a way that we could actually now use it for medical applications and so many others. Generally, what we can understand of coordination chemistry, we can actually see with some examples that we have over here. These are examples of coordination compounds. Generally, this is a paradox actually, because I'm not completely, but I'm blind color, and generally coordination chemistry is very colorful. So coordination chemistry generally is the chemistry of color for these cases of these compounds. So one of the things to explain actually helped to develop some of the orbital theories to explain the color of these compounds and so many other properties, not just the physical properties, but also the chemical properties of this compound. You could actually see over here, we can actually have the center is represented by a cobalt atom. These ones over here, these green balls that we have, they are chlorides. And these are the ones that we have a representation of ammonia molecules over here. If you could actually see that when we have these molecules that they are coordinated, that's the way we say, they are attached to the metal, they are coordinated to the metal, we call it ligands. And these ligands, of course, this is going to be, in this case, this is a ligand, these are neutral. In the case of the chloride, of course, the chloride is an ionic. Of course, then just to be in line with the charges that we have, you see this is positively charged, this has to be a cobalt 3. So one of the other things we can actually, another feature that we can actually identify on these compounds is the fact that they acquire perfectly very well-defined geometries on these compounds. So the cobalt over here is surrounded by 6 different ligands, and these ligands are ordered in an octahedral manner. The octahedral geometry that we have for these compounds can actually see that on the basis. This is, for instance, the equivalent to have 2 square base pyramids, where you have the square base constituted by these two ammonia molecules and this ammonia molecule and this chloride. And of course, the apical position is occupied by this chloride, and the apical position of the other pyramid, which it would be on the bottom, is occupied by this ammonia molecule, in this case. So for this case, also the conformation of the nomenclature that we use in this case is a cis molecule. And this is assigned for the case of the two chlorides over here, that they are occupying different positions and they are ordered, or they are grouped in a cis position. In this case, you can actually see that this compound, although they have exactly the same formulation that we have here, the same constitution, because we have 4 ammonia molecules and then we have 2 different chlorides over here, then we have still the covalent, the center, the 2 chlorides that we have over here, they're opposite one to the other. So in a way that we could actually call for this, the conformation is in a trans conformation to the difference to this other one. Of course, the difference in these compounds are completely different, because one of the compounds is pink and the other one is blue. This is one of the things that this Alfred Werner could actually explain and could actually put in a way that he could actually understand this, first the physical properties. Another thing that we could actually say that for the ligands, in this case, for instance, is only coordinated, or only one donor atom is attached to the metal center. In this case is the nitrogen, and this case is the chloride. This we call it monodentate ligands. Both the chloride and the ammonia, they are monodentate ligands. In this case, for instance, I can actually extend it where I have this. In this case, this is a CH2 and this is a CH2. This is an ethylendiamine, where we have 2 different nitrogens over here, and the 2 nitrogens are coordinated directly to the metal. If you see, I try to keep actually the same conformation that I have for the other compounds, where I keep the 4 nitrogens, which now in this case 2 of the nitrogens are attached by a hydrocarbon change. The same happens here for the ethylendiamine that we have on this other side, but in this case, they are still attached. You could actually see that this molecule is pretty much the same. The constitution is actually exactly the same on this one. But what you can actually see from these molecules is that if you compare this one with this other one, they are mirror images in such a way that they are not superposable. They are not superimposable one to the other. That means that these 2 compounds are chiral. So they are enantiomers one to the other. For that, for instance, the representation that I have for here, the EN, it represents for the ethylendiamine ligand. And in this case, because you have the 2 nitrogens attached by a hydrocarbon chain, this is a bidentate ligand. Of course, if I have 3 atoms for the same molecule, it's going to be a tridentate ligand. It might be tetradentate, it might be pentadentate, as we are going to see later on in some other examples for applications in for medical purposes. In this case, I have the 2 chlorides, and these 2 chlorides that I have over here, they are, of course, occupying a cis position, as it was the case for this very same example that I have over here. But in this case, I have this other symbol over here, which is a delta. This delta represents that this enantiomer is actually the dextro enantiomer. What I mean by dextro, the dextro is only the thing that it turns out the polarized light in a polarimeter in such a way that it makes it turn around to the right. In this case, I got a lambda, which is actually the level enantiomer, that it turns out the polarized light to the left. So you could actually see they are very nice properties. And of course, all this chemistry, if you understand, it's at the beginning of the 19th century. Most of these things was correlated, was compared actually with the properties of the organic compounds. So, but with the plus that now we have a metal. So, and if you could actually see one other thing which is important, we have chirality here, but not in the same sense as we have on a regular chirality for a carbon atom that we could actually have, or regularly as we conceive the concept of chirality, we need 4 different substituents on a carbon, for instance. In this case, you could not actually predict immediately that you could actually have chirality for these 2 compounds. Now, moving on these other ones that I have over here, I have changed already the type of coordination that I have. I have removed one of the ammonia to change for another chloride over here. And this is just to explain another concept in terms of the nomenclature that we could actually have. What have actually left unmoved is actually the geometry is still octahedral. still two different square base pyramids, one here at the top and the other one on the bottom. And now I have three different chlorides on one side and three other different ammonia on the other side. If you can imagine that you can trace a line from here to here, and then from here to here, and then from here to here, this occupies just one of the faces of the octahedron. And that's why, because that is why these compounds are called fascial, or and then the nomenclature for this is FAC in this particular case. So the three chlorides are occupying one of the faces, and this other one are occupying another of the face of the octahedron. In this case in particular, I'm going to be referring just to the ammonia molecules, that all the ammonia molecules are exactly in the meridional position, and on the middle of that base actually represent the base of the pyramid, that you could actually have one here, and the other chloride, which is occupying the other apice of the square, and then this other one. And you could actually see, because they are in the middle, they are meridional, and of course the nomenclature over here is going to be changed by mer in this case. So this is pretty much what we learn when we take a course of coordination chemistry. Of course, all these compounds, they represent a variety of different kind of colors and of course different kind of properties. One thing which is important that I want you to remember is this part of the chirality that can be induced by different kind of ligands, and the fact that one ligand, if it's just attached by a single donor atom to the metal, is called monodentate, and these other ones, because they have two different donor atoms, this is a bidentate, and we will see another tridentate and tetradentate a little bit further on. Until now, for instance, for all these compounds, you could actually see that they are nitrogen, and I said this can be sulfur, this can be phosphorus, this can be oxygen, and so on. When I move what we call organometallic chemistry, the main definition or the original definition of organometallic chemistry is that the compound might actually have, well, no, it must have at least one carbon metal bond. That's going to be the characteristic, and that's why, I mean, we are not really very much ingenious to put names to the chemistry. So in this case, we are using organic chemistry and a metal. So that's why it's called organometallic. So it's really pretty much simple in this case in particular. One of the big properties or one of the big difference on these compounds is the fact that the metal bond is actually very strong. So this carbon metal bond is actually very strong in such a way that these compounds can actually withstand very high temperatures. That's why most of these compounds are used for catalysis. But nowadays, we have learned that the robustness of this compound, it can actually be also used in medicinal chemistry in such a way that can actually not only withstand very harsh conditions in catalysis, for instance, but also can withstand and can really very well stand the conditions for physiological media in such a way that what we want for these compounds is to actually get into the body, for instance, and after that get released or get expelled from the body in pretty much either the same way or in a way that we cannot actually cause accumulation of heavy metals into the body. So in this case in particular for these compounds, and actually they have a very, very high impact, particularly in organic chemistry. We will see some of the prizes, Nobel Prizes, and some of the applications that these compounds have had along the time, but also in material chemistry. Nowadays, many of these compounds are used to make these materials which are called MOFs, metal organic frameworks, that actually are changing also the way we do catalysis and so many other different processes, and also for green chemistry and different kind of stuff, including medicinal chemistry. In this particular case, I wanted to put faces to the names. This gentleman is Mr. Sir Geoffrey Wilkinson, actually. He was a professor at the Imperial College in London. He was actually teaching organometallic chemistry. He's famous actually for one of the catalysts. He actually has his name. He's the Wilkinson catalyst, but he actually got the Nobel Prize because he was actually able to provide a proper explanation of how this compound was formed, and explain in terms of molecular orbitals why this is an iron atom. And on the top we have two slices of cyclopentadienyl, one on the top and another one on the bottom. And sure enough, these kind of compounds are called sandwich compounds. This compound is ferrocene, Sure enough, you're going to be familiar with these compounds, and you can see there is one tab on the top and the other one on the bottom. And in the middle, the filling, it would be actually the filling of iron in this case. This is going to be very important. Well, it's actually very important for different areas of chemistry. For instance, it's a reference in many experiments in electrochemistry, and it's going to be very relevant for some of the design of some of the metallo drugs that I'm going to be showing later on. This is just to present a kind of a timeline on the organometallic chemistry. Many people, sometimes when I presented this, many people think this is a new area. As you could actually see, it's as new as the 1700s. Where many of the story about the organometallic chemistry started actually curiously with some cobalt salts, and for compounds like this one, which is called cacodyl, which is one of the iconic compounds, organometallic compounds, which they have arsenic in its structure, but also some other compounds which are very common catalysts, for instance, like the sea salt. The one who actually was discovered at the beginning of the 1800s, and it was one of the first examples of an olefin, a double bond actually coordinated directly into a metal center, which is something very important for the time, and it was very representative for many of the catalytic process that we study nowadays. This should be very, very known also for the organic chemistry. This was a reaction for the preparation of organochlorosilines for Charles Friedel and Mr. James Kraft, that they actually provided the Friedel-Crafts reaction. And that's also organometallic chemistry. Another one, the Mr. Ludwig Mond, for the discovery of the nickel carbonyl, that nowadays is one of the most studied compound as a fragment, because it's a moiety that it can actually provide a lot of information regarding the electronics and the aesthetics of a given ligand in organometallic chemistry, which is actually very relevant for the understanding of catalytic properties, and also nowadays in medicinal chemistry. You will see that also at the beginning of the 20th century, almost in 1899, we have already the discovery of the Grignard reaction, or the Grignard reagents, that nowadays, I mean, there are many reference books on this, that they are like this thick, and you can actually find an infinite number of applications for the Grignard reactions, different kind of transformations, and several other applications that you could actually have with these compounds, which are very interesting based on magnesium. Of course, we have now in the beginning of 1900s, Paul Sabatier, which very recently also the university in France actually celebrated more than 100 years already. And it was the discovery of the hydrogenation reaction. Hydrogenation reaction has actually affected or influenced many of the common life in our days, not only the industrial, since the industrial point of view, but for many, many other things, for instance, in the food industry and so on. So, and then after that, we are moving in 1909, Paul Henrik introduces a compound which is called salvarsan, which is once again based on arsenic. And then you will see that at the beginning the cacodyl also has arsenic, and this compound also has arsenic over here. It's not rare. I mean, as we know that many of the compounds which are toxic, it really depends on how we do the administration of this compound, and of course the amount of this compound that we provide. But for instance, this was one of the beginnings of the, let's say, one of the very first organometallic compounds that they were actually used for medicinal chemistry. In this case, this compound was used for the treatment of syphilis, particularly in England. And this compound, we're going to see a little bit more later on the talk, how it was administered to people. Now, for the beginning of 1912, it was a recognition already that the Grignard reagents, they were actually very useful for chemistry or for organic synthesis. So the Nobel Prize was awarded to Victor Grignard for the discovery of the Grignard reagent, and to Paul Sabatier for the hydrogenation reaction. And then, of course, you might know the name of Henry Gilman. It might actually ring some bells, because he actually provided the line for the production of the Gilman reagent, which are lithium combined with copper, and these compounds are also very useful for organic synthesis. The ferrocene in 1951, the one just I presented to you, I introduced it to you. This compound had actually been modified in so many different ways, and now there's still a platform, a kind of a organometallic platform, that it can be actually modified in so many different ways to work since material chemistry, regular organic chemistry, organometallic chemistry, and also pretty much now what is interesting to us today, which is medicinal chemistry. We're now moving to 1963. And again, another Nobel Prize. In this case, it was for the reaction for the Ziegler-Natta catalyst, which are metallocines also. And this, we actually owe many of the production of the plastics, the plastic technology to the production of these catalysts, particularly for the production of polyethylene and polypropylene in this particular case, that also from the 60s and onwards have actually changed the way we have the regular life for the presence of the plastics. Of course, nowadays these plastics represent a big problem because we have used it in an excess that nowadays we have a big problem of contamination also with the microplastics and so on. But also these compounds nowadays can actually be used to produce biodegradable plastics, also in combination with some other techniques for biochemistry techniques and so on. So this is the thing that we are trying to do nowadays. We learn from the mistakes we made before, but we're trying to put together nowadays some other solutions for these kind of compounds. 1965, another compound which is also very interesting, which is a cyclobutadiene iron tricarbonyl. This is also a very interesting compound. It looks pretty much like the ferrocene that I have over here. But the thing is, what is important for is because now we have a cycle, a four-member cycle. It means that we have only 4 carbons putting on the top. Let's say we have, let's take one of these things, and then we have a square, which is going to be on the top of that iron, then another 3 carbonyls in the bottom of the molecule. This is very interesting because these molecules regularly, they are not stable by itself. But with these compounds, we can actually make it very stable and they work. Now for the more recent story, which is probably you might know about these reactions, you might have heard about these reactions. Heck reaction, which is one of the reactions which I really love this reaction very much because it has been represented almost 10 or 15 years of the research. We have been decided palladium catalyst for these purposes since I was in the University of Oxford in the late 90s, and then in Hawaii, and then after that we continue with this research back in Mexico. And then we have designed a number of Palada cycles for the purposes of realizing cross-coupling reactions, not only Heck reaction, but also, as we will see later on, some other cross-coupling reactions like Suzuki-Miyaura couplings and so on. Then, of course, in 1973, as you could actually see, Jeffrey Wilkinson, the guy which I introduced, once again, I introduced to yourselves. Jeffrey Wilkinson receives the prize for given explanation together with Otto Fischer to provide the application, not just the explanation of the way the ferrocene is formed, how it's bonded to the metal, and also for the application, the possible application that these ferrocene compounds they may actually have. By 2005, you see there is a very long leap. It's a very long period that it has to pass to again get another Nobel Prize on organometallics in this case. From 1973, where we're talking about another 30 years, where Yves Chauvin, Robert Grubbs, and Richard Schrock got the Nobel Prize for the alkene metathesis, or the metathesis of olefins, for the production of different kind of a number infinite of polymers. And then in 2010 is recognized once again the Nobel Prize for the cross-coupling reactions. And this Nobel Prize goes to Richard Heck and Eiichi Negishi, and of course to Akira Suzuki, that they provide some of the 3 different carbon coupling reactions, which are some of the most relevant for the production of different kind of products, but they have a very big relevance, particularly on the pharmaceutical industry. So putting on this, then you will see these 2 books. I like it very much. You see, they are not necessarily the same name, but pretty much they contain similar information. As you can see, there is not an agreement in this case to say I like this name better, the bioorganometallic chemistry, because it's a combination of all the chemistry with the coordination chemistry, the organometallic chemistry, another word bio, which adds together to this area of chemistry, which actually involves the study of these compounds to use for the drug discovery for different kind of treatments. And we are going to see that these compounds are not really strange, and sometimes they are very often used as a palliatives and also as a medicine for different kind of treatments, particularly for cancer. This compound is very interesting, the one which is on the cover of this book. This compound is actually a ruthenium derivative, but this compound is got a parathymine a moiety on the top. Is again a metallocene derivative, and over here there is also a rutenocycle over here on this part. This generally is a nitrogen, and of course this one is a ruthenium carbon bond. This compound is also well, is a catalyst for transfer hydrogenation reactions, but this compound also works really well for the treatment of cancer. So we will see some of those examples later on. As you could actually see, you can probably know, we'll find it as bioorganometallic chemistry, but you can actually find this area of chemistry as bioinorganic medicinal chemistry, this particular case. And these two books, they are really beautiful on the way they treat, not just from the recent advances, but also from the historical point of view. And I like this, medical square that we have over here, where we have different metals, and their group there is not a casualty that they are organized in this way. For instance, gold, platinum, and ruthenium. There are those two compounds. Well, those two metals, platinum and ruthenium, they are used very often for the treatment of cancer. The same happens with gold nowadays, particularly because these compounds are also very stable on physiological media. But also gold is also used for the treatment of arthritis, for instance. While these other ones that we have here, boron, rhenium, and gadolinium, gallium, sorry, these are used regularly for MRAs or imaging techniques in medicinal chemistry. And the same happens with these ones we have over here with silver, technetium, and gadolinium, which are also used for imaging in medicine. That we are going to see some of the examples, and also we are going to explore, of course, some of the structures of this compound, and of course we are going to see also the design behind these molecules. Of course, as any other area of chemistry, let's say organic chemistry, inorganic chemistry, in the case of bioinorganic chemistry, we have the aid of different kind of areas. In this particular case, of course, we have to make the synthesis, as is stated over here. We make the synthesis, we make the plan of the design of a particular molecule based on the properties that we wanted to see on these compounds, and of course, the target for a particular disease that we wanted to explore. And then once we do the synthesis, of course, we have to make the analysis of these compounds to see that effectively they have the structure that we thought from the beginning. And then we use spectroscopy. We can actually use infrared spectroscopy, UVB spectroscopy, nuclear magnetic NMR, and so on. And of course, we can actually use, if the crystals actually grow in a proper way, we can actually use crystallography. You know, single crystal diffraction experiments are also very nice, because we could actually have the structures like the one represented on here, where we could actually see the way the molecule is actually presented in a three-dimensional manner in the solid state. And we could actually make some kind of interpolation on the solid state to the way it is going to behave in solution. Then, of course, depending on what we wanted to study, we can actually get a lot of help from molecular biology or from enzymology or from any other areas where we could actually see if we can use this compound to kill some bacteria, to kill some microorganism, which is bad for the human being, or if we have a particular disease, let's say once again, let's say cancer, and then it depends on the type of cancer we wanted to explore, and so on. So there are many different areas that it can be combined in this other part. But regularly, the basic steps for the production of the compounds are this one, and then these two areas can actually be expanded quite a bit. So from here, of course, many of the periodic table can actually be used, but so far, what it has been explored, particularly for elements that are considered essential for the body, are the ones in red. So you will see that, for instance, we have, of course, iodine, which is important for the thyroid gland. And of course, we have chloride, we have fluorine, which we have already associated his importance, for instance, for the teeth. We have oxygen, we have sulfur, for instance, which is very important also for the hair and for the growing of certain kind of proteins. We have phosphorus for the bones, nitrogen for the endocrine system, and of course even silicon and carbon and some other compounds which are part of the transition metals, which are the ones I like it very much because they behave really well for the formation of coordination and organometallic compounds. But for instance, in this case, of course, they are considered essential. For instance, iron, if we have a lack of iron, then we have to provide it because otherwise we start to have problems in nutritious problems. Of course, the molybdenum and chromium and magnesium, they are associated also with different kind of vitamins. The cobalt, for instance, is vitamin B, and the vanadium, for instance, which is presumably essential for this, is associated, for instance, with metabolic syndrome, for instance, for the case of diabetes and so on. And that's why so many of the coordination and organometallic chemistry nowadays is pursuing different kind of structures of vanadium to try to see whether these compounds can actually work well for the treatment of diabetes type 2. Then, of course, we have another element that they are pretty much common, like, for instance, magnesium, calcium, once again for the bones, potassium and sodium for the pump of sodium and potassium, which actually regulates a lot of the pressure and the blood pressure in the body. For instance, we could actually see here that the lack of these compounds, for instance, the lack of calcium, of course, retards and also affects the growth of the skeleton, in general, the bones. Magnesium generally is contained in many of these energetic beverages that you can actually take after make some exercise, and it's actually to avoid the muscle cramps. Iron, as I said, I mean, the lack of iron actually causes anemia, and of course, this also affects the immune system. The lack of zinc actually affects a lot and provokes skin damage, and so many other problems that it goes into the body, like for instance, retarded sexual maturation in this case, and of course the copper. It weakens the arteries and it causes liver disorders and so on. And of course, it's associated also with anemia because some of the metabolic routes for the iron and copper are pretty similar. In the case of magnesium, it causes infertility, for instance. You can actually read the other ones over there. But for instance, in the case of the molybdenum, it causes retardation of the cellular growth. Cobalt also anemia, pernicious anemia, which is actually worse than the one caused by the lack of iron. And nickel, they might actually cause depression and so on. The chromium, as I said, is associated with diabetes symptoms and so on. So there are so many of the functions that these metals they actually take into the body. And then what we have is some of these elements can actually be used for the design of novel compounds or the design of novel metal drugs or metallic drugs that we could actually design based on the properties that we already know for these compounds. For instance, one which is been covering a lot of relevance nowadays, precisely because we have actually stood so many times already locked together at home, is a lithium, for instance, that is used as an antidepressive drug. And of course, as I was saying, for diabetes type 2, so many of the compounds that can actually be used, for instance, in this case is vanadium, just to make some mimics, with the insulin, and some other ones which are going to be relevant for different things. And this is the area that we work a lot, particularly with platinum, ruthenium, and in this case, titanium. And titanium actually was used at the beginning as one of the first anti-cancer drugs, but it's not used anymore because it causes a lot of side effects. So many of the chemistry has been concentrated nowadays already in platinum and ruthenium, and I'm going to show you later on some of the best that we have. But some of the other elements that have actually covered a lot of relevance nowadays is iridium, for instance. Iridium has been used very, very much, as well as rhodium sometimes, but iridium has been covering a lot of attention. It's been calling a lot of attention nowadays for the design of anti-cancer compounds. Another ones, for instance, for diagnostic, in the case of diagnostic agents, for instance, for magnetic resonance of image, or MRA, and also for X-ray, of course, some of the lanthanides, and of course barium is also used for these ones. Radiopharmaceuticals was part of the chemistry that I was actually involved when I was working at Essex and also at Oxford University, particularly for the synthesis of rhenium compounds and technetium species. I was not involved in the synthesis of the technetium species, but rhenium compounds, I synthesized some of them because generally to understand the chemistry of technetium, because technetium, generally we use technetium 99M, is radioactive, so you cannot actually work it very simply in the lab. So you generally do first the rhenium chemistry, and if the rhenium chemistry works, generally the technetium chemistry works exactly the same way. So that is why first you have to understand all the chemistry of the rhenium species, and then just try to work directly with the technetium 99M. In the case, for instance, and as I mentioned before, the gold compounds, they are used as anti-arthritic. This is a very well-known kind of compounds. But of course, what is being changing along the days is also the design of the structures, also to have more biodisponibility, bioavailability of these compounds, and also to provide better and more robust structures to actually eliminate the heavy metals from the body. Another ones which are very, very useful nowadays, even nowadays, is, for instance, derivatives of mercury that we know is also carcinogenic. Generally, if we break a thermometer and it's got mercury in it, it's always a big deal on the laboratory, even on the laboratories at the university schools. But for instance, mercury is being used for quite a lot. I'm going to show you some of the structures in a bit. And of course, the silver, for instance, silver is also a very nice compound which has been used for a long, long time. As an antibacterial and as a disinfectant for different kind of wounds. For instance, the silver thiosemic carbacids and stuff like that. So this kind of compound is you might actually find it in the pharmacy, very, very cheap even, and very effective in the way they work. We have also over here antimony, and we have bismuth. Antimony is also been working, has been using for some time, and if you remember, perhaps the toothpaste tubes, there was a time and that they were actually made from an alloy of antimony. Nowadays, they are already made of plastic, but in the case of bismuth, for instance, they might actually be very familiar. You had acidity, a very strong acidity in the body, but it's to avoid the production of ulcers. If you know this, it's a medicine, it's called Pepto-Bismol. Perhaps you know that one. That actually contains a lot of bismuth, and of course it's based in a redox process, and we will see a little bit later on. And of course, there is some other compound, like for instance the fluorine, that they have different kind of properties. Iodine, for instance, which is included already on the salt, the iodidated salt that we consume. That of course that's to avoid problems like another sickness like bosentan, and some other kind of problems for the inflammation of the glands. Then over here, what I wanted to show you, because this has also been a subject of very much attention nowadays, because people now is very much involved on the design of vaccines, for instance. So now you have to know when you can have a beneficial effect of a given compound, or when you don't have that beneficial effect because of the concentration of a given compound. In this case, for instance, we generally will have this kind of curve where we are increasing the concentration of the compound that we have to the point that we have the therapeutic window, or we can actually observe the therapeutic behavior that we want in this case. Otherwise, if we go over that amount, then we are going to start to have toxic overdoses, and we're going to start to see a lot of side effects. We might actually see some of the therapeutic effects that we want, but then after that, we are going to have a lot of problems with the toxicity of the compounds, because either accumulation of the heavy metals or accumulation of the compound itself, or because this compound is actually provided in very high doses. So what we want to do is to actually have it in the proper amount all the time, and of course for that we require a lot of biological studies. In this case, of course, I have to bring different phrases. One from Paracelsus that we are talking about somewhere around almost 500 years ago, when Paracelsus was saying that all the things are poisons, and for that there is nothing, doesn't have poisonous qualities. It is only the dosage that is important for this. In a way, the other phrase that I have to apologize because this I forgot that this is in Spanish, but is you might actually be familiar with Professor Albert Hofmann. Albert Hofmann is very well known in chemistry because he was the one who not only discovered lysergic acid, LSD, but also try it. So he tried it on himself. So he did a lot of experiments on LSD. He wrote a number of books. And he says the difference between a poison and a medicine and a narcotic is only the dosage that we have. So of course, the Paracelsus was used to say nothing is a poison, everything is a poison. It depends on the dosage. So that's why we have to be careful with that. Then another thing that we have to consider, this is a very simple way to explore the way that we can actually introduce a medicine or a drug into the body. As you could actually see, because I mean, everyone has actually taken a medicine at some time in his life. And for instance, in this case, we can actually have it, we can actually take it orally. And then this compound has to have different kind of properties. First, the compound has to withstand the acidity of the body, of the stomach. And the other thing, that's why sometimes we put some kind of vehicles or we come upon some other compounds that may actually work as additives. And in that way, the compound can actually withstand the conditions of the acidity of the body. Actually enough just to travel into the bloodstream and then actually reach the part that we want. So if this actually works well, then the compound actually can go through the membrane to the bloodstream. And then, of course, it might actually have, once again, travel to the membrane, getting to the target site that we want. It might, as I said, I mean, we are still not able to produce drugs that are actually smart. We are working on that. Many groups, many research groups in the world are working on designing these smart drugs, actually to be associated with some other kind of properties using supramolecular chemistry and nanoparticles and stuff like that, to actually get into the target that we really wish. Otherwise, it goes to the target, but also it might actually go to other different kind of targets, and we have to evaluate the side effects that we have over there. In the case that we could actually make the introduction of the drug by injection, then that goes immediately to the bloodstream. We can avoid the problem of that the company might actually be decomposed by the acidity of the stomach. But then again, once again, it has to go to the membrane, and then it has to reach the target site. And then once again, we have to explore the potential side effects that this compound might actually have. Once it happens, once it does its work, this actually it has to be excreted from the body. And the excretion of this compound, it might actually go straight to the liver, to the kidneys. Many times it might actually go to the lungs. Some of these compounds will go to the lungs, those which are volatile. And if it goes to the kidneys, it has to go to the urine. You will see that when you take a medication and when you take any other kind of drug, generally the change in the urine changes in color quite a lot. Generally changes, it might actually go, depending from the kind of drug, it might actually go from yellow to very, very deep yellow. But there are other drugs, for instance, that it changes to red, for instance. So many compounds that they're using as anti-inflammatory, for instance. These other compounds, once you go to the liver, for instance, it might actually go, as in this case, if the compound actually doesn't withstand the conditions, might actually go directly to the intestines, and then of course the excretion go to the feces. So the mechanism is actually looks very simple over here, but of course it might actually be more complicated because the compound to reach the target site is actually a very complicated, very complex process. This I just wanted to mention because it's also a subject that is becoming very familiar for most of the people. Now also thanks to the process that we have had on the news from the vaccines. As you will see, there are different phases for the approval of a drug, phase 1, phase 2, phase 3, and phase 4. Generally, most of the drugs or the vaccines that we have nowadays for the COVID-19, many of them already in phase 3. Regularly, I don't see that many of them are actually reaching the phase 4. And I'm going to mention just very quickly what these phases consist in.
Professor Morels, I'm sorry to interrupt. Can you finish it off in 10-15 minutes time so that we
Yes,
have
indeed.
time? Yeah.
Very well. So I'm gonna try to skip this a little. So I just gonna mention this, that this is a phase 3. This is the number of volunteers. Phase 2, of course, this increases the number of volunteers, and of course we wanted to see side effects and so on. And then phase 3, which is most of these phases are just increasing the number of volunteers that you have actually to explore the efficiency of the drug and so on. Once this thing actually reaches phase 4, most of these things actually goes for selling. It goes available for the people. Most of these things, of course, is for the global trial participation. Of course, these are statistics to 1916. The report for 2020 is about to come out. As you could actually see, many of the people who actually produce the drugs generally are in Europe, and another ones generally where they actually make a lot of these tests are in Europe and also in the United States. And that is actually not red, because many of the companies that actually do new medicines, they are situated in this place. This is, I'm just going to skip a little. This is just part of the things. In this case, for instance, none of this is valid because, I mean, nowadays what we want, for instance, in the case of the vaccine for COVID-19, is that the vaccine actually works. So many of the people is actually participating, and they are really very eager to actually get into that point. Let me get into the cancer. I'm going to mention some of the most notable examples of these metal drugs. Probably you are familiar with this cisplatin, for instance. In this case, cisplatin, for instance, this compound actually was produced. I'm also a wine lover. That's why I put this wine over here to say to serendipity. In this case, it was a casualty, actually, the fact that cisplatin was actually discovered. Barnett Rosenberg, this guy actually was the discoverer of cisplatin, and the experiment that he was doing, he was trying to explore the effect of electricity on the growing of different kind of bacteria. And he was using platinum electrodes, and he was using a buffer of ammonium chloride. So the fact that he actually did an electrosynthesis of cisplatin, and the fact that he used the electrosynthesis of the electricity to produce this compound actually caused that this conformation was produced, because generally the most stable compound for this, or the conformation for this compound, is the trans conformation of the cis conformation. With this discovery, Rosenberg actually provides us with one of the most efficient metallo drugs for for cancer. Nowadays, is one of the main drugs that is using is also because it's cheap in comparison with those which are produced by biochemical processes. And of course, as you can see, it's a very simple molecule. This very simple molecule nowadays, the people has understood that because these two ammonia, they have to stay perhaps into the molecule, because the mechanism for production of cisplatin, the way it coordinates to the DNA, regularly eliminates these two chlorides, and of course it coordinates to the DNA, avoiding the replication of the cancerous DNA, and in that way killing the molecule, killing the cells in the end. You could see that they are different modifications. These two compounds are actually patented, and they are used both in Asia and also in the United States. These two compounds are also approved already in the United States, and they work pretty well for different kinds of cancers. And these other ones, they look very similar, and you say, you could see that the molecule is actually getting more complex, inclusive introduction by the introduction of chiral centers, like in this case. And on this other case, for instance, also there was the exploration of the different oxidation states of platinum, being on this kind of compounds, only platinum 2, and in this case, platinum 4. What is being found is that platinum 4 actually reduces to platinum 2 in physiological media. So in the end, it doesn't make any sense actually to put on making platinum 4 compounds, because they are going to be back to platinum 2 species. You could actually see this ruthenium compound, which is actually very similar to the one I mentioned. I'm sorry to return too much, but this one, particularly this one right here, which is the one I wanted to mention and to show you that this compound is actually very interesting, and actually some of the structures which are very well studied. In this case, for instance, we have an ionic species, going to be more soluble in polar solvents, but also we can introduce phosphates which are empathic or very compatible with water. So these compounds are also going to be very interesting they are being studied. This actually in phase 3. for the studies against cancer. These other ones, which are also very interesting, but then you put chromophores on this part, so you can actually have the effect of light also to do, for instance, photodynamic therapy for the treatment of different kind of cancers. So the derivation, as you could actually see, from a very simple molecule to the ones we have over here, it has been an evolution of perhaps something between around 30 to 40 years. So in this case, this is the molecule. This is actually the presentation that you could actually get. Generally, this is in an intravenous, is administered in an intravenous manner, or bien by injection. And this compound, I'm just going to put a quick video. Let me see if I can put this thing. I don't really know whether you could actually have the sound, but let's try. Can you listen?
Sound is not there.
But,
sound is not coming, but yes, we can see the visual.
okay, let me see. I think that there is a thing with the configuration in here. Let me see. No, I think that you have to actually make it from your side. Well, anyhow.
Yeah.
Let's see the video. So what I wanted to show you here is the way how the platinum actually incorporates into the DNA. As you would actually see, this compound is actually more useful for testicular and ovarian tumors. So the first thing that it has to happen, it has to be a transport, a membrane transport of the compound into the cell. Once the transportation, which might be actually active or passive, once the compound is inside, then let's say the magic starts, how this compound is actually works. So you see what it will happen is the two ammonia molecules, they actually stay, the two ligands actually stay, but the chloride actually get eliminated to produce vacant sites that they can actually coordinate to the guanine moiety into the DNA. So that is going to be the driving force for this compound to actually work, because once it coordinates to the guanines, then actually locks, and when the RNA transcriptase come by, the replication of the DNA cannot happen. And you will see, for instance, that this protein actually serves like a wrench. And what it does is for molecular recognition with this phenol right here, actually avoids that this recognition here actually avoids the replication of the DNA, and eventually the cell dies. Then what it happens is, of course, because it's going to be depending on the concentration of the cis platinum. But if we put too much cis platinum, then it starts to accumulate, and it causes kidney damage, liver damage, and several other side effects that we could actually see when the people is actually submitted to chemotherapy, like for instance the lost hair and so on. But for instance, I'm going to go very quickly, 3 or 4 slides more. Over here, this is the salvarsan, and you could actually see that at the beginning of the century, this was actually used. This is the structures of the salvarsan that they are believed that they were used as a therapeutic for to treat the syphilis, in this case, and these were the kind of syringes that they use it. Of course, this goes to the urinary meatus, and then after that, they have to make an injection, and this pretty much what it does is actually burn the tissue. These compounds that we have over here, you could actually see that we have ligands, which are tridentate ligands in this case, and the kind of method we're using is gadolinium. Most of these things are used for diagnostic in MRA. The same happens with this technician species, that these compounds actually targets the heart, for instance. These other ones where we have carbonyls over here, this actually the carbonyls, the design of the molecule is for these compounds actually to make it soluble in water or physiological media in such a way that may actually work pretty well. Some of the other compounds, for instance, as I was talking about the arsenic, we are actually always considered as a poison. But for instance, this article is very interesting because as it was considered the arsenic, the poison of the kings, is also the savior of the syphilis. If you are interested in reading this paper from the historical point of view, I can actually send it to the professor Carwell, and then probably she can actually pass it to you. These other compounds, for instance, the gadolinium species, which are very, very much interesting, you could actually see these are, most of them, these are polar fragments, like this carbonyl or this amido compounds that we have over here. And these compounds, for in case of gadolinium, actually permits or allows that we could actually have this kind of images, for instance, for magnetic resonance of image. In this case, for instance, to avoid aneurysms, in this case, or in this other case, actually to identify injuries that might actually have in the brain or any other part. I wanted to call to your attention, for instance, that the brand of these metallo drugs in this case is General Electric. As you could actually see, not any other brand for pharmaceuticals. General Electric actually has also his medical section, and it does because it's General Electric Healthcare, and it's because they actually produce the equipment for the MRAs or the resonance for the body. So these are general design for these compounds. If you have, for instance, phosphates in this case, generally we can actually study the bone structure, or we can actually study the brain. If we change this one, for instance, for amines or amides or any other nitrogen combination or sulfur, we can actually study the liver, kidney, and so on. But generally, this is the kind of a structure that generally we can actually study for this. What we want is to have a compound which is very well encapsulated, let's say, very well ligated by the ligands in such a way that the metal is not going to be lost in the process. So in the end, it might be actually excreted from the body very easily. And in the end, these compounds actually might serve for different things. And this, I just wanted to mention one more. I think I'm still on time. For this one, these are porphyrins, and these are very interesting because they have photochemical properties. This, if you could actually see, this property looks very much like the chlorophyll. The chlorophyll, for instance, instead of having magnesium or having iron, in this case, we have palladium, and these compounds, they work really well for the photodynamic therapy, which very quickly it's like, well, these are the structures in the case of the hemoglobin in this case, and in this case, the porphyrin. But for instance, what it does is, and this works really well, is, for instance, the injection of these ones, either by injection or intravenous manner, generally takes a little bit to get into the bloodstream. And once it gets into the bloodstream, this is a very common, is glaucoma, is very commonly produced by high pressure or by people who has diabetes or diabetes type 2. Generally, it's injected, and many times they lose the vision because they have a lot of blood veins which are broken in this case, and because they are broken, the eye actually loses its capacity for the vision. And then in the end, what we have over here is this thing, the laser that is putting on this here actually cauterizes these two vessels over here. This is actually approved. It works very well. And of course, there are many other applications that we could actually see. However, the time I think is over. Isn't it, Professor?
Yeah,
Right.
because of paucity of time, I request you to kindly conclude, because then.
Yeah.
Yeah.
Yeah, sure enough. Just give me 1 minute.
Yeah. Sure. Yeah.
I wanted to show you this a couple more slides. These are also very interesting, particularly for countries like ours. For instance, in the case of malaria, These compounds, which are derivatives of, this is the fragment of the quinine, but you could actually see that we have a ferrocene, and this has actually 40 times the power of the regular quinine, and it works actually really well. And this, for instance, like the sulfate acid of silver, which is the campanile, which is an ointment, which you could actually buy without prescription actually at the pharmacies. These other ones, which are based on antimonium, which are used for leishmaniasis, which are also very well studied and work really well. And there is many, many other development on the research of these compounds. These arsenic compounds, which is also for the this is not leishmaniasis, but this is caused of the sleep, which is caused by the mosca tsetse, but the tsetse fly. And this other one, like the bismuth, this is the Pepto-Bismol. I think is pretty much the same name all around the world, but this is for the treatment of ulcers in the stomach. And these other compounds of bismuth, which are actually, because the antimicrobial activity that they have, they put regularly in tissues, and it might actually cause some, serve for the healing of wounds caused by burning. And then, I mean, we could actually move different ones, but one which I wanted to see, well, these are the gold drugs that we have over here. Generally, the gold drugs, this was one the first ones, and this other one also includes already one sugar, which is very much well assimilated by the body. Or these other ones, I just wanted to show you that you might actually have, or you must have, compounds which are water soluble groups in this case, and this other one too. And this one, which is very interesting. This compound is what we call, or what we know as methylate. Methylate was very popular over here in Mexico because when you get bitten by a fly, generally you get this bump. And to avoid the itching, you get these things put over here, but also to avoid infection. And also this depends a lot on the concentration of the mercury. Nowadays, the methylate actually just gave the name. It doesn't have mercury anymore. The Methylate name as a brand actually is also very well accepted nowadays. And compounds like the ones which are on the top, they are for the treatment, the potential treatment of diabetes, which is also one of the big problems that we have as cancer around the world. Now, with this, I wanted to conclude. Probably in another occasion, we could actually talk a little bit more further on some of these compounds. But as you might understand, as to some of the explanation in the beginning about the coordination and metallic chemistry was necessary to understand this. Thank you so very much again for the invitation. I'm really sorry I couldn't actually go faster, but you could actually see that this thing is, the team is actually very, very broad. So
yeah, Professor Morales, like I'm getting messages from people in my WhatsApp that so nicely explained. So we are very grateful to you, and definitely after this pandemic is over, we would like to invite you to Amity University. And I will just, before requesting our esteemed panelists to say a few words, who happen to be a doctor also, and you have covered so many, right from anti-cancer to anti-leishmania, and I think it will be like for COVID also, you'll be able to, provide some kind of lead. So before that, I'll just ask you one question only due to paucity of time. This is asked by Dr. Anurag Agarwal. He's asking, I would like to ask that those compounds that are useful in some pathological conditions, as you have shown, but they show some toxicities because of not following the rule of 5, that is Lipinski rules of 5. How to reduce these toxicities, how to design them?
Generally, what we do is we design the ligands in a proper manner. As I said, I mean, we've been working on the design of new ligands and the way the administration is given to these compounds. Generally, what we do is to try to modify the structure of the ligands, for instance, some like this, or more complicated than these ones, just to avoid or to combine the toxicity or the potential pharmaceutical application of a given compound or ligand, or the organic molecule, plus the metal. And for that, we use natural products. For instance, lapachol, for instance, or some other secondary metabolites, for instance, of different kind of plants that we know that they have some kind of, for instance, antibacterial activity. We put one which has antibacterial activity with one metal, for instance, platinum or ruthenium, and we actually use it for both for catalysis and also for medicinal purposes.
Yeah. So I think people are regretting that they cannot hear the complete 80 slides that you had there, but we will definitely have the second part in few months to go, and
Sure
then we
enough.
can invite you, because I think today everyone has understood the basic, the concepts, and going further. So I request our esteemed panelist, Dr. Nagpal sir, to say a few words about Amity, and sir is also Vice President for Ritanand Baldev Education Foundation. He has got lifetime achievement award in the field of medicine, and he has got many publications, many awards to his credits, and he looks after the complete Middle East operations of Amity University. So over to you, sir, for your for few words. Yes, sir.
Dear Dr. Morales, it was a great learning experience, and I wish we had more time to listen to you with the innovations that we've gone through. You have taken me back almost 60 years when we were told this terminology in our chemistry class, and we are totally shaken as to what can this be. At that time, even our teachers didn't know what about it. But today, I'm sure that all the people present today, the attendees who have listened to you carefully, must have imbibed a lot of knowledge about the value of organometallic compounds, and in both in lifestyle and also in medicine. And we thank you very much for this time that you spent with us. Regarding Amity, of course, it's a very progressive university, which is now spread across 15 countries abroad, and has about 11 campuses with more than 175,000 students and 6,000 faculty. And I'm sure that once we circulate your presentation amongst all the students, you might receive a lot of response with the questions that they might like to ask. I would only conclude by saying that we are grateful to you, and on behalf of our founder president, Dr. Chauhan, and our chancellor, Dr. Atul Chauhan, we thank you once again for your time that you spent with us, and looking forward to having you again. With great pleasure. Thank you very much.
Thank you very much indeed. Thank you very much.
Thank you,
It is my
Professor.
pleasure.
Yeah, thank you, Professor Morales. And as we had discussed, we'll have many collaborations also with you for projects and publications too. So thank you, everyone. Thank you, sir, for taking out time. Sir is also very busy. And Professor Morales, it is 1 a.m. there, and we are very, very grateful to you for joining. You know, at this time, it shows your scientific temper and, you know, love for science. So thank you, everyone. Have a great day. I'll be sharing the video as well as slides after asking Professor Morales, because they'll have copyright issue, but we will be sharing the presentation as it goes live also today on YouTube. So we'll be sharing with you all. And thank you, Rahulji, for IT support. Thank you, Nagpal sir, again for your time. Thank you, Professor Morales. Okay, have a great day. Thank you. We may all leave. Thank you. Thank you, sir.
Thank you very much indeed. Take care of yourselves.
Yeah.
Take care of yourselves. Thank you very much indeed. See you soon.
Thank you.
Bye-bye.
Bye.